Welcome to episode 76 of the Pulse of Physiology. I'm your host, Dr. Jim Davis, and today we're diving into the regulation of digestion, focusing on how the nervous and endocrine systems coordinate digestive processes. By the end of this episode, you'll understand the role of the enteric nervous system, the difference between short and long reflexes, the impact of sympathetic and parasympathetic input, and the actions of key digestive hormones. So let's get started. The enteric nervous system is also called the brain of the gut, or the gut brain. That's kind of fun to say, right? It's a complex network of neurons embedded in the wall of the GI tract that can operate independently of the central nervous system. The enteric nervous system controls motility, secretion, and local blood flow. It also communicates with the central nervous system via the autonomic nervous system. Now, just like many things in the body, the digestive system or digestive activity is regulated by reflex arcs. Short reflexes are triggered by local stimuli, such as stretch, osmolarity, or pH, and these are processed entirely within the enteric nervous system. An example of this would be the presence of chyme in the small intestine stimulates peristalsis and enzyme secretion. Long reflexes involve the central nervous system and respond to external stimuli, such as sight, smell, or thought of food. So, for example, the cephalic phase of digestion, which activates gastric secretion before food enters the stomach. So the autonomic nervous system control, you've got the parasympathetic and the sympathetic input. So the parasympathetic input, rest and digest, stimulates motility and secretion, and this is mediated primarily by the vagus nerve. The sympathetic nervous system, on the other hand, inhibits motility and secretion and ultimately reduces blood flow to the GI tract. Now, digestion is divided into three coordinated phases. You've got the cephalic phase, the gastric phase, and the intestinal phase. The cephalic phase is triggered by sight, smell, taste, or even thought of food. Ultimately, this phase is where we prepare the stomach by stimulating salivation and gastric secretions via the vagus nerve. The gastric phase begins when food enters the stomach. It's stimulated when the stomach is distended or it's stretched out. It gets bigger, as well as by peptides and low acidity. Ultimately, this triggers the release of gastrin, which increases acid and enzyme secretion and promotes mixing waves. The intestinal phase begins when chyme enters the duodenum, and it regulates gastric emptying and digestion in the small intestine. It involves the hormones of secretin and cholecystokinin, or CCK. Now, there are three key digestive hormones. You've got gastrin, which are released by cells in the stomach in response to food. This gastrin stimulates the release of hydrochloric acid and it causes gastric motility. Secretin is released by the duodenum in response to acidic chyme. Secretin stimulates the pancreas to release bicarbonate, and that bicarbonate will neutralize the acidity of the chyme that's in the small intestine. Ultimately, secretin inhibits gastric emptying. And then finally, there's cholecystokinin. This is released by the duodenum in response to the presence of fats or proteins. This hormone stimulates the pancreas to release enzymes, as well as triggering bile release from the gallbladder. Cholecystokinin also inhibits emptying or gastric emptying. So to summarize, the enteric nervous system regulates digestion locally and communicates with the central nervous system. Short reflexes operate within the GI tract. Long reflexes involve the brain. Parasympathetic input stimulates digestion, whereas sympathetic input inhibits it. Hormones such as gastrin, secretin, and CCK regulate different phases of digestion and coordinate secretion and motility. And so that's it for episode 76 of the Pulse of Physiology. I hope this episode has helped clarify how neural and hormonal signals regulate digestion. Come back next time for episode 77, where we'll shift to the reproductive system and start with an overview of male and female structures. Until then, keep questioning, keep exploring, and stay curious.